We're going to take lipids and follow lipids, their digestion, their absorption, and their metabolism through how the body uses them. So initially, mechanical digestion happens of lipids. So whatever food that you consume is mechanically digested, broken up into small chunks, and through this process, the fats are separated out from the rest of the nutrients. And fats, because they're insoluble in the aqueous solution that is your saliva and your stomach juices, will tend to clump together. So you end up with these big sort of globules of fats that are trying to get away from everything else through the process of mechanical digestion in the mouth and in the stomach. Really, chemical digestion of the fats doesn't start until the fats have been released from the stomach into the duodenum. The chemical digestion takes place when the fat globules are released from the stomach. They travel into the small intestine, and in the small intestine, bile is released from the gallbladder or having been made in the liver, and that bile has different parts that are hydrophobic and hydrophilic. So what the bile does is actually surround little bits of fat and allow those big globules of fat to be broken down into small little droplets that are surrounded by a bile coating that allows them to be more soluble in the aqueous fluid that they're present in. The chemical digestion happens from lipases. So there's a whole family of lipases. There are lingual lipases that are actually made in the salivary gland. There are gastric lipases that are made in the stomach. But the vast majority of the lipases that are really working to break down fats are made in the pancreas and released into the duodenum. Most of the chemical digestion is happening there. The majority of the fats that are consumed are in the form of triglycerides. So a triglyceride molecule, if you remember, is made of a glycerol head group and 3 fatty acid tails. The lipase enzymes take the triglyceride and make it into a monoglyceride by cutting off 2 of those fatty acid tails. Those can then be packaged up with other fats still in the duodenum. So you end up packaging the monoglycerides that have formed from lipase, cutting them off, any fatty acids that are around, with fat-soluble vitamins, cholesterol, any other non-polar sort of substances that you've consumed, get bundled up together into a little fat droplet now called a micelle. These micelles are coated in bile acids still, so that they remain sort of soluble. And the micelles, because they're smaller and because they're pretty soluble, can actually cozy right up to the intestinal villi. They get through sort of the liquidy, aqueous mucus coating on the duodenum and can get down in. And then you remember the process by which non-polar molecules get across a plasma membrane, because a plasma membrane is, the great majority of it is non-polar, so they can just use simple diffusion to get across the membrane into the cells lining the duodenum. Within those cells lining the duodenum now, those micelles get broken apart and they reform with some proteins in, these are the simple columnar epithelium lining the duodenum. They get reformed into a lipid core with some proteins around the outside called chylomicrons. Chylomicrons can then exit from the basement of the simple columnar epithelium. They're too small to get into the capillaries of the bloodstream where the sugars and the amino acids get. So they're big enough that they end up mostly in lacteals, because the lacteals have flaps that will open and allow the larger fat molecules to get in. So they get taken through the lymph system, eventually get dumped into the bloodstream, and they travel around delivering, the chylomicrons now are traveling around delivering triglycerides, for the most part, to cells that need fats. Eventually, though, those chylomicrons will end up in the liver. In the liver, they get reprocessed into more lipid covered, so there's a lipid interior with a protein exterior. In this case, they're called VLDLs, or very low density lipoproteins. So the liver will allow these very low density lipoproteins to get released into the blood again. The very low density lipoproteins have quite a bit of triglycerides that they're still delivering to fat cells, but there's a lot of cholesterol in them. So after they've dumped off their triglycerides, they're mostly cholesterol. So there's a cholesterol core with proteins around them, and now they're called LDLs, low density lipoproteins. These low density lipoproteins have a pretty bad rap because the more cholesterol, higher the cholesterol level in your blood, that cholesterol is in the form of LDLs. So an LDL is cholesterol going through your blood trying to be delivered to cells. And it just keeps flowing around in your blood trying to be delivered to cells unless another molecule that the liver makes, a protein coat called an HDL, or a high density lipoprotein. The HDLs go out into the bloodstream and they're picking up cholesterol to get rid of it. So this is why there's a strong correlation between high LDL levels and bad cardiovascular problems, because it's high cholesterol and it's cholesterol that's in your blood trying to be delivered to cells, whereas high HDLs tends to be correlated with good cardiovascular outcomes because it's actually picking up cholesterol and trying to get rid of it out of your body. So fats, we've talked about digestion, we've talked about absorption, now let's talk about how fats are used. So if your body needs to use fats, the dietary fats tend to be stored in adipose tissue. In adipose tissue, they're stored as triglycerides. So when your body needs energy, it takes those triglycerides and uses a process called lipolysis to break down the triglycerides, and they in fact get broken down to glycerol, which is half a sugar. So it can go straight into the glycolysis process to make ATP. Fatty acids, on the other hand, don't have any hydroxyl groups. They're straight up nonpolar carbons. So they take another process called beta oxidation that converts them and allows them to enter the cellular metabolism process more in the Krebs cycle area. In the process of beta oxidation, ketogenesis also happens. So some of the fatty acid parts get turned into ketone bodies. These ketone bodies can be used by cells. They can actually feed into Krebs cycle as well to make ATP. But in diabetics, where they can't actually use glucose because they don't have insulin that allows their bodies to take up glucose, those ketone bodies can actually lead to problems. And because so much fat is being used, because the glucose can't be used, so many ketone bodies are made, it can actually put them into ketoacidosis, which can actually lead to shock and comas and kill them. So the food that we intake can be converted into carbohydrates, amino acids, and fats. Those fats could be burned for energy. They could be stored in fat cells. They could be turned into other things we need, like hormones. And your body needs fats to build more cell membranes as well, so they could be used structurally in the body. So there's a lot of important reasons for having fats in your diet. So when are fats used and when aren't fats used? The time right after you eat a meal for the next about 4 hours is called the absorptive state. It's assumed that you have a fairly balanced diet and everything you ate wasn't fat. So theoretically, there's enough amino acids and glucose in the food you ate that pretty much during that time right after you eat, the main fuel that's used is glucose through cellular metabolism. So any fats that you consumed in this time, you don't really need for ATP because your body's using glucose anyway. So you will tend to store those fats during the absorptive state. That absorptive state is actually modulated by insulin. So when you eat, theoretically that food you ate had carbohydrates of some kind. Those carbohydrates will trigger your pancreas to release insulin. The insulin triggers glucose to be used, and any excess at all will be stored. So insulin is pretty much triggering the use of glucose, the storage of fats. Adrenaline will actually turn off that process. So epinephrine or adrenaline is the opposite in this case. Post-absorptive state is, okay, now I haven't eaten for about 4 hours, so now my body does not have a lot of blood glucose just immediately available that came into my intestines. In this particular case then, the body will take triglycerides that are stored in fats or that might still be circulating in the blood and do the lipolysis process to turn them into glycerol and fatty acids and use them to actually burn ATP. So the post-absorptive state is when you're actually using those fats to burn for energy. This state is controlled by lower blood sugar levels, and that stimulates the pancreas to release glucagon. Glucagon, in fact, will stimulate the process of fat breakdown to allow for it to be metabolized. In addition, adrenaline will do this, growth hormone will do it, cortisol, thyroxine. So there's a lot of hormones that will help this process of raising the blood glucose levels by taking stored nutrients and turning them into things that can be used in cellular metabolism. The other part of the control process really for all of this is the homeostasis of when do you feel hungry and when do you feel satiated. That's how you end up in the absorptive or the post-absorptive state. Hunger can actually be controlled by the presence of fatty chyme going into the duodenum. That fatty chyme will actually decrease the contractile force of the stomach and the rate of stomach emptying. Theoretically, the fuller your stomach is, the more hormones it's sending to your brain saying, I'm full, don't eat anymore. So there's actually some diet theories that you should eat a little bit of fat early on before you eat, the idea being that then you've got some fatty chyme getting dumped out of your stomach, pre-telling your body to not eat so much that you're full. I don't know whether that really works or not. The other two really important hormones in hunger and satiety is that the release, so the stretch of the stomach is one thing that will release hormones that tell you you're full. The other is insulin. Insulin will actually trigger leptin release, and leptin is really the hormone. It's made from adipose cells. So adipose cells, as they get full and as blood sugar is high, it will actually go to the hypothalamus and say, I'm full. Don't eat anymore. I'm feeling good. The opposite of that are hormones, either low blood sugar or a hormone called ghrelin will actually trigger hunger, say, I need to eat more, I'm really hungry.